going to open and kick off today's forum. So good afternoon. Firstly, my name's Suman Majumdar. I'm a infectious disease physician. I work at the Burnet Institute and I'm a fledgling PhD student at the Department of Paediatrics. I'd like to first begin by acknowledging country. I'm speaking from the lands of the Wurundjeri people and pay my respects to elders past, present and emerging. Today, the forum is going to focus on environmental health and we've got a very, I'd say almost niche, but very exciting series of talks and topic around water quality, environmental health in Fiji, which is an area of focus for many people at the Melbourne Children's Campus with projects in other health issues in Fiji. So there will be great interest. I know we've got people coming in online, but this forum will be recorded and also shared to many others. So we have two speakers today. We have Professor Karin Leder and Dr. Aaron Jenkins, and they will both present and then we'll have time for questions through the chat function at the end. In terms of announcements and housekeeping, I just wanted to flag that there is the 3-day Centre for Research Excellence Pneumonia Symposium next month. That's in the newsletter. And there's also the next forum around that same week in November, which will be on strengthening health systems with Saya Abnabola presenting on, which will be a great talk. So I'll first like to introduce Karin, who's an infectious disease physician, and she's a co-lead of planetary health and the head of infectious disease epidemiology at the School of Public Health and Preventative Medicine at Monash. She's also the director of research for Revitalising informal settlements and their environments, or the RISE program, which will be the topic of today's talk, and also the head of travel medicine and immigrant health services at the Victorian Infectious Disease Service at Royal Melbourne. Karin has been involved in diverse areas of clinical and public health research, looking at the risks of infections from exposure to contaminated environments and unsafe water, and factors associated with the spread of infections across borders in travel and tropical medicine. So thank you, Karin, for joining us from Suva today, and looking forward to your talk.
Thank you so much for having me. I'm just going to get my slides up. You're seeing that right in the right forum?
Correct,
now?
Yeah, that looks good.
Great. Look, thanks so much for the invitation to present. As you said, Suman, I'm in Suva. My first time back since COVID, and it's really it's a great time to be talking about RISE, because I'm in Suva for RISE, and I couldn't be more enthusiastic than I am when I'm actually seeing the progress on the ground. So RISE, as you mentioned, is a program of work revitalising informal settlements and their environments. And I want to give a brief overview of what is a really massive and transdisciplinary, and I think really exciting area of work. I'll only be able to touch on the surface of many bits of it, but it's RISE itself is a randomised controlled trial, and I'll run through what our theory is, what our intervention is, and what the assessment is and where we're up to. So firstly, where is RISE working? Well, it's in urban informal settlements in 2 countries, firstly in Suva, and we are deliberately working in communities that are affected by flooding, either tidal flooding or pluvial flooding, and many of them living in conditions like this, with particularly in the wet season, very damp environments, and any you can imagine that any pathogens that enter the environment from improperly managed sewage systems or open defecation can be spread all through communities because of the flooding and leading to exposure and potential illness in the children. The second country that we're working is Indonesia. We're working in South Sulawesi, in Makassar, again in similar conditions of informal settlements. And you can see the difference and the problems with access that people will have in getting into their houses in the wet, particularly in the wet season. So what we're wanting to address is water and fecal waste contamination, but also reduce exposure to any contaminants in the environments that you can imagine people would be exposed to if they were wading in this kind of water. This is a similar picture from one of our sites in Makassar during the dry and then the wet season, where you can see that, you know, access and therefore potential exposure to any contaminants are a real problem. So what are we actually doing? Well, a fundamental premise of RISE is that there are 1 billion people that live in urban informal settlements, and this is, you know, this number is rapidly increasing. And despite our ambitions, there are still 2.3 billion people that lack adequate sanitation, and more than 500,000 children under the age of 5 that die of diarrhoea annually. So the vision and ambition of RISE is to improve human, environmental and ecological health in urban informal settlements through a novel approach to water and sanitation management that will hopefully reduce environmental contamination and also reduce exposure through our interventions, such that any contaminants that are residual in the environment are less likely to come into contact with the kids, or the kids are less likely to come into contact with the contaminants, and therefore will improve their health. More specifically, our research aims are reduce fecal environmental contamination and assess the impacts through intervention that we call water-sensitive revitalisation, or nature-based solutions, and I'll explain what that is in the next few slides. But our intervention will change the built environment such that we will either give better access pathways and drainage to reduce that kind of flooding, reduce that dissemination of pathogens in the environment, and therefore improve the human health and the intestinal environment of the children who live in these conditions. And the intervention will also reduce, by improving water and sanitation management, will it reduce pathogen contamination of the environment, improve the ecological and environmental health of the environment through this nature-based solutions approach. So what is nature-based solutions? Well, traditional approaches for water and sanitation have either involved big pipe solutions such as we have in Australia, but they're expensive, and they the people who live in these informal settlements are decades, at a minimum, decades away from having access, for governments giving them access to this kind of solution. The other approach that has been used is WASH, water, sanitation and hygiene, where often new toilets are put in, but and people are given education and potentially better drinking water quality, so to improve their hand hygiene and reduce ingestion of microbes. But unfortunately, recent WASH trials have not achieved the extent of health benefits that public health was hoping for. There are billions of aid dollars put into WASH, and there are improvements in children's health, but because the impacts in some recently performed randomised controlled trials have not shown the extent of health impacts that people were hoping for, there's now an understanding that we need to be thinking about WASH Plus, which is essentially the same kind of systems, but thinking about the environmental contamination. Just putting in a toilet or just telling people, giving people better drinking water is not going to reduce exposure to contaminants if the environments that they live in are still contaminated, or if the septic waste from the toilets that are installed are not properly managed, because otherwise environmental contamination will still continue unabated. So if I can get this to work, the approach that my colleagues in engineering and architecture, oops, sorry, I might not be able to get this to work, on using is essentially to capture grey water from kitchens and showers and put it through biofilters, as well as black water from toilets that goes through pressure tanks and then septic tanks, and then all of this gets put through first subsurface wetlands and then surface wetlands before it's released into the environment. Doesn't matter if you don't understand it all, but essentially it's using nature to take septic waste and grey water waste, using a little bit of smart technology in the pressure tanks that we will be installing, but essentially using nature, using soil and in subsurface wetlands and then surface wetlands to filter contaminants such that any waste that is produced and that is released into the environment has gone through 3 layers of treatment in septic tanks, the subsurface wetlands, and then the surface wetlands before it's discharged into the environment. And through this technology, which, as we've said, mainly relies on nature and is very easy to maintain, and communities can be very much engaged and can be handed over the management of these simple systems, we anticipate that we'll be improving water and sanitation management and reducing environmental contamination. In addition to these systems, where in some of our communities we'll be installing rainwater tanks and toilets where, if and where it's needed. Sometimes toilets are already there, it's just that the septic waste from the toilets need to be managed. And sometimes in some places we are putting in hand basins, etc. as well. So our approach is very much to design what's needed according to our assessment of each site that we're working in, to see where the pinch points of exposure and contamination are in the environments, and then to co-design with communities how any toilets and basins, so we're putting them in wet pods, where how they and where they should be installed, how and where any tanks should be installed, and in particular how and where any of these nature-based solutions should be installed. So to show it kind of in a bit more life in one of our demonstration sites in Indonesia, you can see how these different elements can come together, with potentially tanks being installed, a walkway to elevate people or to improve people's access to their homes, underneath which is a whole lot of technology, and on the side of which is subsurface and then surface wetlands. Sorry, I've gone the wrong way. This is showing this in a different form, but essentially in this site, an elevated walkway was needed, with pipes and other things that are needed for our engineers installed underneath the walkway, and our subsurface and surface wetlands put to the side of this, which actually adds greenery. It makes places look nicer. And in the demonstration site in Indonesia, you can see the same environment before the installation of our intervention, where people are clearly wading through potentially contaminated waters, and then afterwards with this elevated walkway that's given access, but also these treatments, these nature-based treatments that just make it look nice. It looks like, you know, gardens on the side, but they're actually filtering the contaminants before the water is released. We have finished a demonstration site in Indonesia, and on the 1st of September of this year had what was known as a groundbreaking ceremony. We're about to start the build in the in the communities that we're working in. I'm in Fiji at the moment because tomorrow we're doing an opening of our demonstration site in Fiji, and this doesn't look as beautiful as the what I showed you in Indonesia, and that's only because I'm going to the demonstration site tomorrow and haven't been able to update my pictures yet. But essentially, now that I've explained the intervention, I want to explain a bit about the research and the context in which we're installing this intervention. The intervention is, as described, this nature-based approach that we're installing in a site-specific way in 24 informal settlements, 12 in Makassar, Indonesia, and 12 in Suva in Fiji. And the research that we're aligning to this installation as part of the randomised controlled trial is to look at what the impacts will be in the sites that have got the intervention versus those that are initially going to act as control sites on the ecology, on the environmental health and environmental microbiology, and on human health. And we're doing this via an unblinded randomised controlled trial. Our primary outcomes are to be looking at the impact of the infrastructure on pathogen presence in the faeces of children under 5 in the communities that we're working in, and also to look at elements of their microbiome and antimicrobial resistance, diversity and abundance in the children's species and also in the environmental specimens that we're taking. It is a complex program where we have different teams. We have a design and engagement team. We have a team of people who are involved in taking and analysing the specimens from the ecology and the environmental specimens. We've got human health sampling and surveys in kids under 5, health and wellbeing in the broader community, and then a program of work around policy and upscaling. But in terms of the randomised controlled trial, what we are doing and what we have done is to do a series of baseline pre-randomisation surveys and sampling regimes in each country, in each of the 24 settlements, then randomly allocated the 24 settlements into 12, 6 in each country, that are going to receive the intervention initially. We are then going to do 2 years of post-build monitoring, and then the other 12 sites, 6 in each country, are going to be upgraded. As I've said, there's too much to talk about to go into great detail, but just showing you some pictures from the sites. Lots of work going on in environmental sampling. Here, some of the team with their eskies, because I presume they've taken some feces and they've got them in the eskies to take them back to their lab. Surveys in the homes of the communities, water samples, which are being taken. We do all our sampling on Survey CTO. We've got our houses barcoded, all of our samples barcoded. So all the data is captured in a de-identified way. We then have this complex, I guess we call it the wheel of life, but it's this complex series of campaigns where we take it in turns to get into the field and to take bioacoustic recordings of nature, temperature and humidity recordings, mosquito sampling for our, what we call they're the 3 elements of our ecological assessment. We then take water and soil samples and something which is called boot socks, where our field teams put like surgical boots on over their shoes and walk dedicated transects of the communities, and then we look at what pathogens we found on the boot socks. We're taking grab samples of soil and water, and that's our environmental sampling. And we're doing a whole range of analytics on it. And then from the health and wellbeing, as I've said, we take feces from children under 5. We do health surveys of both kids under 5 and more broadly of the older people in the household, including economic well-being, social, emotional well-being, as well as physical health. And then for the children under 5, we also do annual blood sampling and anthropometrics. So we started this program of work in 2017. Like all programs, we have been interrupted by COVID, but to give you an idea of the scale, you can see we have just under 600 households in Indonesia and just under 900 in the 12 communities in Fiji. You can see the number of people. Since we started our baseline surveys, we've had an increase in the number of households and participants in all 24 of our settlements, slightly more in Indonesia than Fiji, but that's reflective of lots of different things, including the increase in the populations coming into urban informal settlements. What I'm really proud of is that despite the fact that our intervention has been delayed, and I'll show a timeline on the next slide, we have incredible ongoing survey participation, and there's a somewhat approximate numbers because we're still doing surveys, and in fact we're doing this survey right now. It changes a little bit with each round, and it changed a little bit during COVID, but also I think really importantly, ongoing equally good participation in both sites that have been randomised to get the intervention kind of nowish, and those that know that they're not going to get it for at least another 2 or 3 years. Give you an idea of, we're taking lots of things in our surveys, lots of different symptoms, but you can see, the level of diarrhoea in kids under 5 in Indonesia and Fiji respectively. The orange bars are kids under 5, the yellow bars are older children, and bottom 2 graphs are fever. But, that we're not seeing a whole lot of drop-off in the reported rates of diarrhoea if we were or fever that you might expect to see if we were getting a lot of survey fatigue. And you can see pretty high rates, around 10% or so of diarrhoea in Indonesia and kids under 5, a little bit lower, more around 5% in Fiji. Lots of work going on in the lab. In fact, again, one of the exciting things about this program is that we have set up labs in Makassar and in Suva to do this work. We have incrementally equipped these labs with more and more sophisticated technology, including now quantitative qPCR to do our pathogen work across all our different samples. It is all our local teams in both countries have now been trained in mosquito identification, a whole lot of analytics, E. coli counts, and also pathogen detection, and we're moving into genomics now. So, we're just reliant on our absolutely awesome teams. And I'm going to skip over this very quickly, but I don't expect you to read it, but our multiplex pathogen detection is able to detect, over 40 different pathogens simultaneously. And again, it probably doesn't predict very well, but the different colours here for each of the pathogens is what we find in human, soil, boot socks, water, and animal. We're taking animal feces, animal scats as well. And so even if you just look at this, very quickly, this is a really unique data set that is now, being able to look at pathogens and also genomics, and we will be looking at AMR as well from the AMR abundance and frequency in the samples that we have, but, across this whole different type of sample range, which is one of the unique things. We're also monitoring the exposure of the children, because it's not just what's in the environment, it's also what kids get exposed to in the environments. So we were lucky enough to get some additional funding to do some videography of the children to see where they play, and we can map some of, where do they usually go in the environments in their communities, so where are they likely to be exposed. We're mainly looking at the outdoor environments because, that goes along with our improvements in fecal management, but we also recognise that there's other routes of exposure, including in indoor environments, and we'll be looking at that as well. As I've said, again, really quickly to skip over, to show you, we're getting heat data, indoor and outdoor heat data. We're getting mosquito data, in both countries. We're getting this bioacoustics or noise data. Don't ask me to explain this slide in any detail, but it's just this fabulous transdisciplinary network of researchers who are collecting different data types longitudinally, but also in this randomised controlled trial so that we can see the impacts of the intervention. The original timeframe was a 5-year project that was meant to be finished a couple of months ago, from August 2017 to July 2022. We got a little bit behind, admittedly, even before COVID, as I think most big projects do, but we did collect baseline data, longitudinal baseline data, pretty much over a year, because that's really important in the environmental context. In 2018 and 2019, 2018 in Makassar, 2019 in Suva. And in each country, we had these really exciting randomisation ceremonies that were very much led by the communities. The randomisation was done in front of the communities, and it was a real highlight. And we were very lucky that we had got these done, prior to COVID hitting. We then did have a period like many programs have where we couldn't move forward as we had anticipated because of COVID. There was still a whole lot of lab infrastructure development and training and remote survey work and designs from our engineers and architects that was going on in the background. But obviously the intervention couldn't start because we couldn't get into the field safely. We did construct that demonstration site in Makassar prior to COVID. So we now actually have a couple of years of monitoring of the water that's going through that demonstration site, and we can see that it's doing what it's meant to be doing in cleaning the water. And as I've said, I'm in Suva now to open our demonstration site here tomorrow. So we'll be starting the construction. Well, we've had our groundbreaking ceremony, as I said, in September of this year in Makassar. So they're really shovels are hitting ground, now-ish, but in Fiji it will start next year. We hope to finish construction, if not next year, then early the year after. We then need to have 2 years of post-construction monitoring, all these same samples and surveys, etc., continuing in what will then be intervention and control communities. And then when we finish the 2 years of monitoring, we'll construct the intervention and upgrade in the control sites. Our main sources of funding, and there are many partners, but for the research side of this, our main funder is the Wellcome Trust. And for the infrastructure, we had some initial funding for our pilot site from the Asian Development Bank, but our main funders are GFAT in Makassar and MFAT, which is New Zealand Aid in Suva, and also Monash University. So it's a massive consortium, and I'm grateful to all my colleagues and all our funders and all our partners, because this will now be, at least an 8-year program of work that we think is really exciting, and that we hope generates the evidence to help inform how we can better manage water and sanitation across informal settlements for the most vulnerable people in the future. If you want to know more, we do have a website, and I will stop sharing there.
Fantastic. Thank you, Karin. That was a real power of work, and you can really see how much there has been in terms of collaborations and partnerships to get there. I do want to hold over questions to the end, if that's okay. We'll get Aaron to talk next, and if people want to use the Q&A chat function, just please punch in your questions as they come, and we can address them at the end. So thanks very much, Karin. So now I'll introduce Dr. Aaron Jenkins. Aaron is a Fijian citizen of Eastern European and native North American Cherokee descent who has lived and worked for decades in the communities of the Pacific Islands. He's Oceania's inaugural Senior Research Fellow in Planetary Health with the University of Sydney School of Public Health. Edith Cowan University Centre for People, Place and Planet, and the Fiji Centre for Communicable Disease Control. Dr. Jenkins is a board member of the Fiji Institute of Pacific Health Research's new Centre for Communicable Disease Research. He's really worked on pioneering integrative approaches like environmental change, linking environmental change to human health outcomes. So Aaron's going to talk about the work he and his colleagues have also conducted in Fiji, which is called the WISH Fiji project. And I'll hand over to Aaron to talk to that. Thank you.
Thank you. Let's see. Yeah, Bulavanaka, ladies and gentlemen. I'm greeting you in a Fijian language as this platform of work really arises from Fiji, and I'm a proud Fijian citizen. I've spent most of my life living and working in Pacific Islands, so it's a great pleasure that I'm able to share with you today the progress of this program of work known as WISH Fiji, or the Watershed Interventions for Systems Health in Fiji program, which has gained some reputation worldwide as an example of planetary health in practice. My name's Aaron Jenkins, or Eroni in Fijian, and I'm a senior research fellow in planetary health with the University of Sydney's School of Public Health, Edith Cowan University's new formed Centre for People, Place and Planet, and a researcher with the Fiji Centre for Communicable Disease Control. Just briefly, the outline of this talk is to first share a little on our oceanic context in terms of our shared and intersecting environment and health burdens, the foundational research and origins of this WISH program, and then to introduce briefly the scope of the WISH work to date. So this place that we are, that we speak of, that's often referred to as the liquid continent, it's Oceania. It's this vast area of some 156 million square kilometers, almost a third of the Earth's surface. And this is Vanua for 41 million people across Australasian, Melanesia, Micronesia, and Polynesia that call this region home. And Vanua is this oceanic wide term in various forms that acknowledges that material, biological, social, and cultural dimensions are inextricably interrelated. And it's also a recognized place-centered model for health and well-being. So we know Oceania has some of the most globally important and unique natural cultural diversity, supporting rich complexes of terrestrial marine ecosystems and the world's most diverse range of indigenous cultures. Papua New Guinea alone has about 1/6 of the world's languages. Oceania more than 1,000. And this vast oceanic region thus has the most to lose, culturally speaking, from the pressures of global political and economic change. But these landscapes and seascapes that support the health and well-being, the culture and economies are delicate, and they're rapidly transforming, reducing resilience to both environmental and climate change. Continued habitat loss and degradation, overexploitation, invasive species, pollution, and a changing climate are undermining the ecosystem functionality and service provision in these island ecosystems. Primary health threats in the region relate both directly and indirectly to these rapidly changing socio-ecological settings, particularly infectious diseases relating to water pollution and poor sanitation, zoonotic diseases related to large numbers of subsistence livestock and animals and informal animal husbandry practices. And the urban transition is also central to the NCD crisis, with obesity and diabetes rising disproportionately faster in this Pacific region over the past 3 decades than in the rest of the world. So I'd say an early point for me for WISH coming must have been sometime around 2008, after another bout of severe flooding and heavy rain in Fiji. And my wife, Karin Jenkins, who many of you may know, was working on typhoid vaccination in Fiji at the time after a series of outbreaks. And she said to me, you should really watch, and 6 to 8 weeks after this flood, we're going to have typhoid going up around this flooded area. And sure enough, this turned out to be the case. And we see in Fiji and many Pacific Island countries that flooding following cyclones and prolonged rainfall is linked to outbreaks of a variety of waterborne bacterial diseases like typhoid, leptospirosis, and Shigella. Here you see in 2012, Fiji was impacted by flooding from sequential tropical depressions that caused this widespread flooding. And these events resulted in substantial population displacement and significant post-disaster increases in leptospirosis cases, Here you see 3 to 8 weeks following each of the major flooding episodes. So in 2011, I became involved with this interdisciplinary team of researchers and practitioners that was under the banner of Wildlife Conservation Society's HEAL, or Health and Ecosystems Analysis of Linkages, or HEAL program, which has evolved into the Planetary Health Alliance. And around this time, we produced this model of how the combined effects of watershed modification and more intense cyclones and heavy rains are creating this perfect storm of conditions that leads to outbreaks of water-related disease and negatively affects human well-being through combined impacts on both food and water resources. So we tried pitching this idea to a consortium of donors in order to address, promote more integrated management and plan for multiple benefits. But unfortunately, we didn't receive funding at the time, but I decided this was too important to let lie. So I went off to do a PhD to study this problem. And in short, our studies and those of our colleagues showed that amplified typhoid and leptospirosis cases was associated with vegetation removal and erosion in watersheds, proximity to flooded areas and livestock, and that upstream watershed conditions in rural areas also predicted childhood diarrheal disease. So this suggested that the evidence to guide both the natural and built infrastructure interventions in watersheds is therefore critical for water and sanitation management, as well as other downstream ecosystem services such as flood mitigation during adverse weather events. And speaking of adverse weather events, in February 2016, Cyclone Winston hit Fiji and caused massive damage to drinking water and sanitation infrastructure and to watershed systems. And there was lots of outbreaks of typhoid and leptospirosis in the months to follow. So the government, the coordination and prioritization of the resources and of response was really hampered by this lack of integrated health, sanitation and water quality information. And so with some funding from WHO and UNICEF, I worked with the Fijian government to start creating a system which could integrate drinking water quality and sanitation facility monitoring with health risk surveillance systems to enhance the recovery and response. This all began well and generated some good intersectoral willingness to coordinate information, but it all petered out as the cyclone response funding dwindled. And soon afterwards, with generous support of the Australian Department of Foreign Affairs and Trade and Bloomberg Philanthropies and some additional funding from the Bill and Melinda Gates Foundation, we're able to tie these various threads together of work into the Watershed Interventions for Systems Health Program, or WISH Fiji. So WISH is really grounded in an adapted health system framework that embeds the project in a multi-sectoral perspective, explicitly acknowledging the role of ecosystems in health security. This proposed adaptation of the model conceptualizes governance not as the building block, but as the mortar holding the other building blocks together. This new health security framework provides the structure for our study design with activities in all 3 circles of this framework, in the traditional health systems, in integrated cross-sectoral governance, and in the recognition of the ecosystem services that sustain health. As a way to engage with this complexity, in WISH, we conceptualize natural systems as nested subsystems. There are risks present and opportunities to act within each nested and connected subsystem, from the global climate system to the environmental and social contexts, residential settings, individual behaviors, organs, unicellular organisms, and the genetic mobile elements that work together to regulate health. Interactions between the scales in systems ecology are considered as remembering for these downscale interactions that draw on biotic legacies of large, slow processes like forest assembly, for example, and revolt for upscale connections from smaller scale, faster processes like microbially mediated disease outbreaks, for example. So the primary aims of WISH in this context are to empower communities to access and maintain their fundamental right to clean water, develop a coordinated mechanisms for systems health governance between communities and government and across sectors, strengthen connections to place to enhance environmental stewardship and to maintain cultural practice, and of course, reduce the incidence of water-related disease in people in downstream coral in 5 watersheds in Fiji. Facilitate approaches to sustainably finance and scale these interventions nationally and internationally. So here we recognize watersheds, also known as river basins and catchments, as an effective place-based functional unit that offers a coherent and ecologically representative subsystem in which to address multiple environmental, socioeconomic, and health objectives together, linked to achieving the Sustainable Development Goals. Globally, waterborne disease kill more children than AIDS, malaria, and measles combined, making it the second leading cause of death among children under 5. And furthermore, water-related diseases, which include vector-borne infections, account for 17% of the global infectious disease burden. So Pacific Island countries also now have the lowest coverage of improved drinking water and sanitation on Earth. 2/3 of people rely on unprotected water sources for drinking and cooking. So in which we're particularly tackling the most insidious and widespread of the water-related diseases that Pacific Island countries often refer to as the 3 plagues, which is leptospirosis, typhoid, and dengue. This region, the Western Pacific region, has the highest global incidence of leptospirosis and typhoid, and the second highest incidence of dengue after Southeast Asia. And despite the various efforts to control these water-related diseases, the interventions that focus only on the disease, the human disease, and ignore this greater environmental and social context have really been unsuccessful. So WISH evaluates and addresses water-related disease risk by combining metrics of health, water safety, sanitation, and land use, and emphasizes this need for action research at the land, water, and health nexus. Our project is focused on 5 watersheds in 29 rural communities with high burdens of at least 2 of the 3 plagues. Each of the 5 watersheds are unique. The largest watershed of Wambula on Viti Levu has a population of over 6,500 and is a river that's 32 kilometers long. Our smallest catchment is Beretta on Ovalau with a population of just under 1,000 and the river is 10 k's long. And the forest cover differs between each, but the general pattern is that there's more forest coverage in the top half of the catchment and the most people in the bottom. So it's a participatory mixed method action research, and it includes both the quantitative and qualitative co-production of knowledge alongside local key stakeholders. And we use iterative cycles of planning, implementation, and evaluation. We're using a before and after design to evaluate health and health system outcomes, water quality, as well as social and downstream ecosystem impacts such as coral disease. And at the national level, we're working with key stakeholders in both the design and implementation of the work. Within local intervention sites, we work with the communities using participatory approaches, and the data is gathered to guide the design of the site-specific interventions. So our WISH Fiji journey has almost closed one loop of this iterative adaptive management cycle. We've progressed over 4 years from identifying our watersheds, then I must emphasize a very long 5-month period of community engagement using a tailored community level free prior informed consent process, designing our sampling instruments, collecting the baseline data, identifying our risks across the various nested scales, designing communication tools to communicate these risks, water safety and sanitation planning in every community to identify the wash risks, and then the intervention planning to reduce the risks and implement the interventions and then monitor outcomes, which we've just done a monitoring intervention after. So this is just to give you an idea of the range of the different 14 survey instruments that we have, from mapping government initiatives to community mapping, household questionnaires on agriculture, fisheries, wash concerns, observational data, and a large set of water and soil samples that are collected and tested. In any event, a massive amount of data is being captured to evaluate health and health system outcomes, including the effectiveness of cross-sectoral and cross-scale processes, water quality, social and ecosystem impacts within and downstream of our intervention watersheds, such as coral disease and the abundance of food fish. Ultimately, we have enrolled a total of 311 households and 29 communities across our 5 subcatchments in our action research. And this is one of our study villages in the Upper Navua River system. We have a large team of around 17 working in the field that have presented all the data back to the 29 villages. And based on this data and local knowledge, water safety and security plans are drawn up for each village, and the specific nature of the local interventions then are determined in consultation with the village committees. And these are a combination of both built and natural infrastructural interventions to reduce the common drivers of water-related and coral disease spread, and also the decline of freshwater and marine food resources. So here you can see in this risk factor table the different nested scales across watershed, community, household, and individual at which risks for systems health are acting, and where they're common for multiple diseases, including leptospirosis, typhoid, dengue, and coral disease. And you can see with the yellow arrows that there's clear efficiencies to be gained by acting on the risks at multiple nested scales and where risks are common in multiple diseases. So here we can modify environmental risks at a number of scales nested within watersheds and be addressing multiple outbreak-prone diseases and downstream ecosystem health. A total of 339 interventions were identified across all of our 29 communities using this place-based participatory process, including several that were identified after the multiple cyclones that went through. And you can see the widespread of general categories, with water supply, improvement, and protection the most commonly identified, followed by agricultural practice or land use management, animal management, drainage, waste management, sanitation, integrated planning, and then health. So a very wide spectrum of interventions, mostly outside the purview of health systems. This just shows you the spread identified across the 5 watersheds, the different types of interventions that were identified. And these are the spread of the 154, about half of what was originally identified that we've now actually implemented. COVID did stall our sanitation installation efforts, but these are now underway, and drainage remediation is also underway, but also not captured in this table. These are just what has been completed to this date, so 154 different interventions. So we pared down from our data to 18 important risk factors that influence 2 or more of the diseases of interest. And this risk matrix shows the risk profile of each village for each risk factor across each nested level across the 29 villages and 5 watersheds at baseline. And as you might imagine, red is high risk, yellow is medium risk, and green is low risk. And the basic situation here is that at baseline, we have these communities where we know lepto, typhoid, dengue, and diarrhea are prevalent to different degrees. And what we see is that 80% of the communities do not have safely managed sanitation, in part because over half of the communities have damaged or overflowing sanitation infrastructure. This, along with a bunch of other key land use factors, is contributing to microbially unsafe environmental water in over 41% of the communities. And about 25% of these communities have highly contaminated primary drinking water sources and the pipe water that reaches the house. And proximity of swamps, uncovered household containers, and poorly maintained community drainage are also contributing significant risk for these communities. Here we have data from baseline marine surveys that shows very high levels of coral disease incidence from our 4 Wish Fiji watersheds that drain to the coast. Coral disease prevalence rates ranged from 8 to 12%, with the average for each subcatchment above the 5% threshold for healthy reefs, suggesting that these reefs were experiencing environmental stress. So using our risk matrix, we can then create dashboards for each community for this set of 18 risk factors, and you see how they're tracking, with the red area indicating a deteriorating condition, yellow indicating no change, and green indicating positive change. But just some brief results. Here's one of the several positive changes that we've seen in post-intervention. If we look at this particular risk related to adequacy of supply of drinking water at a community level across all the communities, the number of high-risk communities has come down and the number of low-risk communities has increased. And in this case, we spent quite a lot of time fixing and replacing water systems, and this appears to be going in the right direction. So access is generally improved across our communities. If we then measure the quality of the drinking water across all the communities, this provides empirical evidence of reduced E. coli contamination, with 7 high-risk communities now reduced to 3 and low-risk communities increased by 3. So this is also going in the right direction, with many of the source waters now fenced off or in protected areas. So not only access, but also quality of primary drinking water has generally improved. Surprisingly, this also extends to environmental water or water from the nearby river or stream, where numbers of, numbers of high-risk communities decreased and low-risk communities increased. So that is quite a surprising result, actually. Certain positive changes in behavioral risks were also notable, particularly with respect to hand washing and personal hygiene. I'm pretty sure that our frequent messaging and supply of wash kits had an influence, but really that COVID was probably the greatest influence here. Additional high risks, including proximity of swamps, uncovered household containers, and poorly maintained drainage, are also contributing significant risk for these communities, particularly vector-borne disease. They remain largely unchanged, and the data is still messy, but I need to point out here that swamp proximity is largely a watershed management issue and doesn't mean we should just go out and drain the swamp. So we also created this bespoke model for based on our data collected on the 29 communities. And the most important variable, if you look at the length of those green bars, was an environmental and landscape factor, which is the proximity to swamps, then socioeconomic and demographic factors like relative wealth and mobility, followed by a mix of environmental factors related to erosion and runoff, and then behavior measures such as removing standing water around the house. So there's firm ground to suggest that this model is correctly specified and would be adequate for predicting purposes and assisting design of interventions to reduce cases of dengue. Important to note that the interventions need to be made from the landscape or the watershed scale all the way through to the individual scale simultaneously in order to really reduce dengue incidence. And this can only really be done through cross-sectoral surveillance and community action. We also have nested within the WISH project, building from our previous work on typhoid in the Central Division. We have a consortium of researchers who worked on 3 of the 10 Gates Grand Challenge grants to work on a better understanding of the environmental niches of typhoid. And in short, we know that watersheds that are impacted by anthropogenic activity result in soil erosion, high sediment yields, and elevated nutrients. And this is associated with high typhoid fever incidence. And we hypothesize that sediment-associated biofilms are an important ecological niche for S. typhi survival. We can see that in sediment biofilm, there's a significant co-occurrence of a soil amoeba, acanthamoeba, and S. enterica. Experimentally, we see that in the presence of this amoeba, which forms cysts in water, typhoid is still able to survive in water after 500 days. Some of the bacteria are free. You can see that in that little picture, with the red arrow. Some bacteria seem to be associated with the cyst, which is the white arrow, and other bacteria seem to be located inside of the cyst. We also found that typhoid genomes rearrange their structure in water, with the most common arrangement being GS 11.1. We also have now confirmed S. typhi in the environment via PCR, and I have to say this is a remarkable result for a number of reasons. For typhoid, this is one of only the first couple confirmed hits of typhoid in the Fijian environment, and one of only a few research groups worldwide that has been able to successfully do this. It's also a great validation for me after saying for years that we need to be more wary of environmental sources of typhoid and consider the likelihood of environmental reservoirs. It took us a while to optimize our PCR, so we still only really looked at 30% of our samples. Right. So we have multiple detections of our microbial pathogens in several of our watersheds, which can help target the remedial action in these communities with some urgency. For the PCR detects, they're both from leached zones of latrines in the Upper Navua and in Dawasamu. And from our metagenomic data from the first 100 deep sequence samples, we also had 5 strong hits for lepto. And by far, The raw abundance is highest in the 3 Dawasamu samples from dam sediment, community drainage, and the water straight from a spring. In Upper Navua, it's in lower abundance, but leptospirosis was present in the pond sediment and also from in-community tap water. Part of our work, so with the metagenomics and part of our work and the PCR is to trying to find these pathogens of interest wherever they are in the environment. And then among all the trillions of types of microbial life, we've been using both PCR and metagenomics to help us pinpoint these. We've only really looked at 1/6 of our 800 plus samples deep sequenced. It's extremely large and complex data set. It's in fact the largest repository of metagenomic data on freshwater systems that I know of in the Pacific Island region, and about 74 to 98% of the taxa within each sample still is unidentifiable. So the discovery scope is immense. There's a whole variety of other pathogens, including Vibrio cholera, leptospirosis, Mycobacterium, tuberculosis, bovis, and they all should be of interest in helping direct public health interventions. Our work also reveals high levels of AMR genes that are present in rural Fijian water and sediment, and it's a real first detailed information available on AMR in the Pacific rural environment and points to a clear trajectory of important future research in the next phase. I might just skip through this one, just in the interest of time. Our first PhD student did publish this paper earlier this year, which shows that weather seasonality and rainfall predict the syndromes associated with leptotyphoid and dengue. In short, the different early warning and response syndromic conditions is associated with a different rainfall lag. And you can therefore possibly to predict when outbreaks will likely occur following seasonality and rainfall patterns. You can pre-position diagnostic and treatment resources that can then be aligned with the spatially explicit seasonality and rainfall peaks to plan and address water-related disease outbreaks. To facilitate the cross-sectoral governance that we need, we took a review of policies, statutory and regulatory frameworks that govern both watershed management and human health in Fiji. And we're preparing a policy brief that seeks to provide policymakers with a rationale for implementing integrated intersectoral approach to watershed management, and identifies issues with current policy practice and provides recommendations for improvements. But the great news is, working with the WASH cluster, we also prepared a cabinet paper, which I'm very pleased to say just passed a few days ago, or a week ago. It was announced by the Fijian Health Minister in the media last week as part of our WASH workshop. And the cabinet directive is a clear example of the Fiji government's commitment to continue to provide a legal platform to provide safe drinking water through evidence generated by integrating water-related disease surveillance with climate information, sanitation surveys, water safety planning, and drinking water quality monitoring. It's really important because this committee is important for health systems governance, because it weaves together human health and the environment in an explicitly linked legal framework, and it also helps bring together health and WASH, which has historically been poorly linked in practice. This all happened during COVID as well, so we had deep integration with our government and communities, which means that we were able to provide several avenues of support to our government and our 29 communities. I'll just skip quickly through this, but
thanks, Aaron. Sorry, yeah, we're getting at time now. So
Fiji has also been through 3 cyclones, so we provided support for that as well. Basically, we need to take this multi-sectoral systems approach, and Swish Fiji is a systems model of cross-sectoral coordination. It efficiently progresses many of the sustainable development goals, but it requires sustained investment in the interventions across the various nested scales to realize the full benefits, particularly for nature-based solutions that exhibit lagged responses. Our work will continue into Papua New Guinea and the Solomon Islands, funded by Kiwa, the Kiwa Initiative. But in the interest of time, I will leave it there. So thank you very much.
thank you, Aaron, and thank you to Karin for 2 fantastic presentations. look, we are right on time because there's just so much content there, so we won't have time for questions today. But thank you, Karin, for answering questions in the chat there. And I think a lot of interest is on the economic side, but also around the engagement and partnerships, which as I indicated before, has got to be immense in both your projects and real a cornerstone of what you do. And I guess it's eerily timely with what's happening in Victoria at the moment with floods in our communities, around the importance of this kind of work and its impacts on human health broadly. But look, I'm really grateful for your time and presentations, and I'm sure you both wouldn't mind being contacted by others from this group if there are questions or follow-up.
No problem.
Yeah.
No problem.
Great.
Sorry,
Okay.
we didn't have leave time for questions, but yeah, no, very happy to be contacted.
Okay. Thank you all. All right.
Thank you.
Good afternoon to you.
Great. Thanks for having us.
Thanks for